Welcome back to MLT 120. This is part 2 for the Intro to Hematology lecture. We're going to be starting off by talking about cell structure. So why would it be important in the field of hematology to know the parts and the structure of a cell? It's because in hematology we focus a lot on cells and identifying what is normal versus what is abnormal, and particularly for our blood cells. So we need to review the parts of a cell. Some of this information may be familiar or a review to you if you've previously had another science course or a biology course where you have learned the structures or the components of a cell. We're going to go through each of the key points or the key structures of the cell. And we're going to start off by talking about the plasma membrane. It is the outermost membrane or the outer boundary of the cell. And you can see where the cell membrane is on this little diagram here. It's the outermost membrane on the very outside of the cell. And it's made up of a lipid bilayer which contains phospholipids and cholesterol. If you look here on the right hand side, you can see a zoomed in or a magnified image of a plasma membrane. So this is your plasma membrane, this whole structure here, and you can see the lipid bilayer. Bi meaning two, the heads are on the outside. There's this layer and then there's this layer. It's actually a phospholipid bilayer. There's also things like cholesterol that's contained in your plasma membrane. Also, something to note about our plasma membrane is it is semi-permeable, meaning it can allow certain molecules to pass through to and from the cell. You can kind of think of it as a barrier or a gatekeeper. The next structure we're going to talk about within the cell is the endoplasmic reticulum, or the ER. There's actually two types of ER. There's the rough ER and then there's the smooth ER. The endoplasmic reticulum is a membranous network of randomly distributed structures throughout the cytoplasm, and it stores and transports fluids and chemicals throughout the cell. The rough ER has ribosomes on its surface, and they produce proteins, where the smooth ER does not have the ribosomes and has a smooth surface, and they produce lipids. If you look over at our diagram, these are examples of endoplasmic reticulum, and it looks like these actually have ribosomes on the endoplasmic reticulum, so this would be an example of rough endoplasmic reticulum. The next structure that we're going to talk about is the Golgi apparatus. And you can go ahead and see the Golgi apparatus right here on our diagram. It's located next to the nucleus and the rough endoplasmic reticulum. It usually looks like a system of stacked membranes or bound flattened sacs, and it modifies and packages macromolecules and other organelles for secretions. So you can think of the Golgi apparatus kind of like a post office. The Golgi apparatus also plays a role in producing granules which are present in our white blood cells. Our next organelle for cell structure are our mitochondria. Our mitochondria, and we can see one right here and we can see one right here, are randomly distributed throughout the cytoplasm, and they produce energy, or what we call ATP, for the cell. And we usually refer to the mitochondria as the powerhouses of the cell. Okay, the next structure is the nucleus. It's usually located towards the center of the cell. Okay, and it can be either round or oval in structure. And this purplish structure right here in the center is representing our nucleus. It's usually composed of DNA and proteins, and it contains the genetic code for the cell. And it also controls the cell's division and the function of the cell. And on the next slide, we're going to zoom into this nucleus and look at some structures that's actually inside the nucleus. Okay, so as promised, we're actually zoomed in and are looking inside the nucleus on this slide. Within the nucleus, we have the nucleolus. It's usually round or irregular in shape, and it's composed of ribosomal RNA. There may be more than one inside the nucleus. If you look over here on the image of the nucleus, this whole structure itself is the nucleus. And right there in the center, this particular one only has one. There's the nucleolus. And the function is to synthesize the ribosomal RNA. Next, let's talk about the nuclear chromatin. It is usually double-stranded DNA. It can be referred to as unorganized DNA, and it's also located in the nucleus. So let's look over here at our image. All this stringy type structure is our nuclear chromatin. You can see this line right here is pointing to all of the chromatin inside the nucleus. And one last thing I want to note on this image before we move on to our other structures of the cell, this nuclear envelope that's on the outside of the nucleus, look at this nuclear pore. So information or molecules can be passed in between the nucleus and the rest of the cell. So just like the cell itself and the plasma membrane, things can pass in and out of the cell, things can pass in and out of the nucleus. Next up on our structures to know within our cell are lysosomes. These are randomly distributed structures throughout the cytoplasm. They contain hydrolytic enzymes and they help in the cellular digestive system. And if you look here on our diagram, we have a lysosome labeled right here, and we also have a couple more represented on the diagram. They're just not labeled. The next structure is the centrosomes. These are located usually near the nucleus, and it usually serves as the insertion point for the mitotic spindle fibers, and it plays a role in cell division. And cell division is also known as mitosis. So if you look over here, we have our whole cell structure, and right here's our nucleus, and you see our centrosomes located right here, and you can see this is a magnified image of what our centrosomes look like. This image is just showing you the overall structures in more of a 3D image of a cell. It also includes some structures that we didn't mention. Some of the structures that we didn't mention are used for locomotion or movement of the cell. This cell is showing an example of flagellum, a tail-like structure that could help the cell to move. Also, cilia could help cell structures to move. But if you want to, I would pause the video at this time and study the structures. And also remember, you have your cell membrane that's made up of a phospholipid bilayer. Now we're going to switch gears a little bit and talk about cellular forces. In particular, we're going to talk about osmosis. Osmosis is the transport of liquid, particularly water, from an area of lower concentration to higher concentration. So let's look at the image in the slide. Remember, when we're looking at this image, we're looking at the movement of the liquid or the water. We're not trying to look at the movement of the little balls that's representing the concentration. We're looking at the movement of the water. So let's look at our before image. We have a semi-permeable membrane represented by this light pinkish color. So on the left-hand side, we have few or very few balls on the left-hand side, and that's representing a low concentration. On the right-hand side, we have lots of little balls representing a high concentration. Also in this before image, notice the water level. The water level is the same. Okay, so this is before osmosis happens. Okay, now let's look after osmosis happens. We're going from a lower concentration to a higher concentration. So all of our water, or a lot of our water, not all of it, our water has moved across this semi-permeable membrane from a lower concentration to a higher concentration. So now look at the level of the water. The water has went up on the higher concentration side because the water wants to go from low concentration to high concentration in osmosis. So why is this information we just learned important for hematology? We must understand how osmosis works and the effects on the cell or the blood cell in hematology, because in hematology testing we need to count our red blood cells. And a red blood cell is basically just an outer membrane filled with hemoglobin. So it's easily influenced by the concentration of a solution that they're suspended in. So in the lab, we dilute down red blood cells often to make them easier to count. So we must know how the solution or the concentration of the solution affects them. The next concept we need to be aware of is tonicity. Tonicity is the concentration of a solution as compared to another solution. And there's three different terms that we need to know and understand that fall under tonicity. There is isotonic, there is hypotonic, and there's hypertonic. Isotonic is when the concentration of the solution is the same on the inside of the cell and on the outside of the cell. This is the state where the cell is the happiest. Next up is hypotonic. This is where the concentration of the solution is lower than the inside of the cell. So let's look at the image and think back about what we know about osmosis. So if the concentration of the solution is lower, the solution on the outside of the cell, what's going to happen to the solution? It's going to want to go from lower concentration on the outside to inside the cell. So what's going to happen to our cell? It's going to swell up. Now finally, let's talk about hypertonic. This is where the concentration of the solution is higher than the inside of the cell. So the solution on the outside of the cell has a concentration higher than inside the cell. So the water from inside the cell is going to go out. So our cell is going to shrink or crenate. So we talked about each of the three situations on the previous slide, but let's talk about each one more in detail for tonicity. So what happens when the red blood cell is placed in an isotonic solution? Okay, if we put our red blood cell in saline, which is about 0.85% sodium chloride, which is isotonic, with our red blood cell, the red blood cell will not change. This is due to the red blood cell and the saline being isotonic. There's no need for there being exchange of water inside and outside because it's the same concentration on the inside of the red blood cell as the outside of the blood cell. So if there's no exchange of water inside and outside of the red blood cell, there's not going to be a change in the structure and shape and size of the red blood cell. So the red blood cell is going to stay the same. So again, when we're looking for a solution to suspend red blood cells or to dilute red blood cells down in the laboratory, we look for a solution that's going to be isotonic with our red blood cells, because we don't want our red blood cells to swell up, to shrink, or be damaged in any way. We want our red blood cells to be isotonic with whatever solution we use. So what happens when the red blood cells are suspended in a hypotonic solution? So let's say we have a hypotonic solution that has a concentration lower than our 0.85. And an example of that would be our tap water. Our tap water is hypotonic. So when we put our red blood cells or suspend our red blood cells in tap water, the concentration of the red blood cells are greater than the solution. Thus, what's going to happen? The water from the solution will move inside of the red blood cells, and that will cause the red blood cells to swell up like a balloon, and they can swell up to the point that they can pop. If they pop, this term that we use in the lab when red blood cells pop is called lyse. And if this happens, we cannot count them. Remember, one of the main things that we do is count the number of red blood cells patients have. So if they lyse or pop, we won't be able to count them. So we do not want to use a hypotonic solution to suspend our red blood cells in the laboratory. And you can see here, you can no longer see the nice little donut shape of the red blood cell because it is swelled up with water from the outside coming into the inside of the cell to the point that it has burst or popped, or the technical term lysed. Now, what happens to a red blood cell when we suspend it in a hypertonic solution? Let's say we have a solution that has a concentration greater than our 0.85%. So when our red blood cells are suspended in a hypertonic solution, the concentration in the red blood cells are lower than the concentration of the solution. So what's going to happen to the water? The water from the inside of the cell is going to rush out of the red blood cell and go to the solution. So what's going to happen to our red blood cell at that point? It's going to shrink up, or it's going to do something we call crenate. You're going to end up with crenated red blood cells. If our red blood cells end up crenating, we can also have problems analyzing the red blood cells in the lab and in the hematology department, so we cannot use a hypertonic solution to dilute our red blood cells. And if you look over here, you can see these little red blood cells were placed in a hypertonic solution, so they're all swiveled up or crenated, where all of their water, or most of their water inside the cell, has flowed out into the solution. All right, it's time for a little trivia to review the information that we have just been learning. An MLT student makes a dilution of patient red blood cells by adding 0.5% sodium chloride. What will happen to the red blood cells? And you can pause the video at this time before we go on to the answer if you want to try to answer it on your own. The next slide will have the answer. Your options are: they will become crenated, or they will lyse, or they will remain the same. The answer is B. They will lyse. A 0.5% solution is less than a 0.85% solution. Thus, the solution is hypotonic. Water will travel from the solution to inside the red blood cell, and the red blood cell will swell and most likely lyse. Here's a little cartoon explaining the difference between a hypo, and an isotonic solution. There was a mad scientist that wanted to destroy red blood cells. And to start off, they put them in a hypotonic solution, the red blood cells. And when they put it in the hypotonic solution, guess what happened? The red blood cells swelled up to the point they were going to burst. So here's our hypotonic solution. Next, the mad scientists put the red blood cells in a hypertonic solution. And guess what? The red blood cells shrunk. And finally, the red blood cells were put in an isotonic solution and nothing happened. They were completely happy. So this is just a fun little cartoon to show you the difference between the three different types of solutions. Again, we want to put our red blood cells in an isotonic solution because that's where they're most happiest. There's not going to be an exchange of water inside and outside of the red blood cell, and that's why we use it for diluting down samples in the laboratory. This concludes our Intro to Hematology lecture. I hope you have learned a lot about cell structure and also osmosis. I will see you over in the next lecture.